I would like to extend it a little bit. My talk will be a little bit provocative, but I'm trying to explore with you where cardiovascular pharmacology will be going in the next 20, 30 years. So I'll analyze with you where do we actually stand, and we have already got a very good critical overview of where we stand. What are the reasons for the current status? And sort of my outlook is what can big data and systems medicine probably tell us and change how we will do medicine in the next decades. So I will review the current drugs, some of the new drugs, the efficacy, some additional points on efficacy that you've heard so far. Also review the failures that have occurred, the preclinical basic research connection to that, and that's the slightly provocative part, our concept of disease that I think we critically need to review. So you all know that it takes about 15 years in average to bring a drug to the market. So from the initial idea of a target, compounds that bind to that target, until the drug actually is registered. And the average cost for a drug to be brought to the market now is $3.8 billion. That figure, of course, contains all the unsuccessful attempts to develop a drug. They basically have to be refinanced by this one successful drug. But in average, that's how expensive it is nowadays. And the costs are actually exploding exponentially. So you've heard about the statins. This table roughly summarizes the drugs that we are currently using for cardiovascular diseases, more or less. Diuretics, spironolactone, and it also shows have there been innovations recently, and are these innovations out of patent? If an innovation is out of patent, it means in the last 15 years there has been no innovation. Otherwise there would be compounds still patented. So you could say eplerenone is not a conceptually new drug, but it's at least an improvement of spironolactone. Beta blockers are all out of patent. ACE inhibitors have been replaced by angiotensin receptor blockers. Renin inhibitors try to get into the market, but they are not really an improvement. Also, this does not represent a conceptual change in targets. It's still the same target. Statins, I would subscribe to that, are essential drugs. Any other attempts then have been rather marginal in success. The PCSK9 inhibitors are, of course, a very interesting development. You may know that there is quite an intense debate on the role of cholesterol as the major component of statin efficacy. That may be answered by another drug that also lowers cholesterol and mortality. All other drugs that lowered cholesterol did not significantly or to a relevant factor lower mortality. So that steered this discrepancy. All attempts to increase HDL and thereby have patient-relevant outcomes have failed. Then we have the nitrates. It's over 100 years. Another vasodilator were endothelin inhibitors. And probably the only real significant innovation were the introduction of SGC stimulators, the receptor for nitric oxide in the moment for pulmonary hypertension. And vitamin K antagonists, TPA, are sort of not preventive drugs. They are try to overcome a prothrombotic state. DOACs are in principle the same approach, but with better pharmacokinetics and probably slightly better efficacy. But in principle, we have not really seen many conceptual advances. We are still dealing with more or less the same drugs, the same targets, the same approaches. And a lot of these drugs are used in very different cardiovascular disease states, as if they would be all related. Thank you. So one discussion about statins is statins act very early in a very complex and important synthetic pathway. About 20 different products are influenced by statins inhibiting HMC co-reductase. So it is possible that what we observe clinically is exclusively due to the reduction of cholesterol. But I think one should always be open for the possibility that the fact that statin regulates so many other mechanisms in inflammation, for example, that it may also possible that statins, the pleiotropic effect of statins, contribute to the clinical outcomes that we observe and explain why other cholesterol-lowering drugs did not have the same clinical outcomes. And that has resulted in now almost disagreeing guidelines between the US and Europe. US basically dropped the term lipid-lowering therapy, and in their cardiovascular or coronary artery guidelines replaced it simply by statin therapy and defined conditions for high and moderate dosing. The inclusion criteria are basically patients with clinical symptoms, patients with diabetes, and patients with a calculated risk factor. So no mentioning about cholesterol levels. There's only one case if LDL-C is above 190 mg per deciliter, then they use cholesterol levels as an inclusion criterion to start statin therapy. That is highly controversial still, but it's at least well based on the literature. The PCSK9 inhibitors may yet again change the picture if they act exclusively by lowering cholesterol. The other attempt was to increase HDL levels because not only high LDL levels correlate with a risk of coronary heart disease, but also low LDL levels correlated with that. But one big mistake that one can make in medicine to assume that a correlation means a mechanistic link. It doesn't. There are a lot of things that correlate but have nothing to do with each other. And insofar, it's surprising, but also a lesson that none of the pharmacological attempts to increase HDL have produced any patient benefit. One reason may be that we assume there is only one HDL, but we know now that there are several forms of HDL. Some are anti-inflammatory, some are actually pro-inflammatory. And that brings me to the point that we are currently too much focusing on surrogate parameters and do not really understand the mechanism of the diseases that we're dealing with. One example is diabetes, where we lower blood glucose, and we know that at least the early generation of blood glucose lowering drugs have no effect on patient-relevant outcomes or mortality. Another example is hypertension. It's similar to the statin story. Several studies have shown that, yes, it's an important risk factor. Yes, it is beneficial and important to lower blood pressure, but we need to critically appraise that the patient benefit that we observe or that we can obtain with that is still very, very limited. And you know that 95% of all patients with hypertensions are classified as essential hypertensives. This in medicine is a very elegant term for saying we have no idea why the patient has hypertension. We give a drug that dilates blood vessels and lowers that blood pressure, takes away that symptom or risk factor or surrogate marker, but we do not treat the mechanism why the patient actually became hypertensive because we don't know the mechanism. So therefore, we are in an area where we do a couple of the right things, but what we do is still rather imprecise. And that's reflected by these number needed to treat, which for severe hypertension are quite acceptable, but for mild hypertension, to prevent one stroke or one MI per year, they're rather high. So classical drug discovery still had this dream of a blockbuster billion-dollar drug. So to develop one drug that one can give to millions of patients and have this one-size-fits-all approach. And we are rather treating symptoms in the moment than mechanisms of the disease, because we don't know the real molecular mechanisms of the disease. But you have to know a molecular mechanism in order to have a molecule against which you develop a drug. We also assume that one drug always has one target, but statins are a good example that maybe statins have 20 different effectors in the end. And the fact that most cardiovascular drugs are out of patent shows you that there really is an innovation roadblock in our field. But not only our field, it's a worldwide phenomenon that the success rate of drug discovery is going down and down, whilst the costs, as I said, are exploding exponentially. Actually, it has been called the E-rooms law. You may have heard of Moore's law. That's that the number of transistors on microchips increases every year by a factor of 10, every 2 years by a factor of 10. So in drug discovery, they have a little bit cynically made the reverse, E-rooms law, that the efficacy is actually dropping every 9 years by a factor of 2, and it's getting worse and worse and worse. So there is a fundamental conceptual problem that we have in drug discovery. And when you look at the reasons for failure, the number one reason failure is efficacy. The drug is just not doing what it was expected to do, based on basic science data or based on very sort of structured clinical trials that sometimes have not so much to do with the real patient situation. Coming back to this, it's also worth looking at the preclinical phase, that basic research that generates the hypothesis for later drug development. And another major problem that we're facing here, which you may have heard about, is that was the first paper by Prince. Now there are 5 papers out of that we have a major reproducibility issue in basic science. So this was from a study that Bayer did. Before they decide they move on to a target to develop a drug, they had sort of the tradition to reproduce 8 or 10 key publications from this field, top papers, just to be sure that they see the same thing that has been published. And after about 15 years, Bayer was opening their archives, and they said, actually, 65% of the published literature we cannot reproduce. And this is top papers in top journals. So no one knows how it is with the sort of not so high level journals. So we have a huge problem also with respect to the knowledge that's generated from basic science and eventually leads, of course, to clinical hypotheses. And one of the major issues here is publication bias. It's understandable because what scientists are usually measured against are papers in journals. Very rarely are they measured against the patient benefit their science has achieved. And there are lots of reviews out of that. I just show you 2 papers from our own work. Don't look at the targets so forth. We're working on stroke targets in the moment. But we could not find, or we could not reproduce what 8 other publications had reproduced. And when we analyzed the data in this funnel plot, it's called, how the published data, these are the open symbols, how do they, are they distributed about the effect size. And we saw that and these data should be distributed like a Gaussian curve. You see, you find in the literature positive data, and then all of a sudden the curve stops. There are no negative data in the literature. You can then calculate how many negative data must be there unpublished just to fill the Gaussian curve, but these data must have been generated unless Gauss was wrong, but they were never published. And another example is here. Don't look at the target, but again, all papers in Nature, Cell, Science on another target in stroke, the same thing. This is the distribution of the data in the literature, and basically here, this other right half is completely missing in the literature. And with this, of course, you generate a bottom line which yields to irreproducible data. And if a company now, actually 3 companies developing this target as a drug, and it's quite likely that the drug will be ineffective because the scientific basis on that is flawed. Statistically also, you know that people very often brag with these data were statistically significant, p smaller 0.05. But there is another number that's equally important. Was the study powered enough to actually calculate a statistical significance? And that's the level of 0.8 power of false positives. And basically none of the data sets was statistically powered to even measure the significance. So it's also a huge quality issue. So where do we stand? Last point is our concept of disease. This is Patrick Wellens. He's the head of GSK R&D, good friend of mine. He also works scientifically in the same area. And he stated that currently many of the disease labels that we are using are 19th or 20th century disease definitions. They are not molecular or cellular classification. And what he predicts is that the better we understand the diseases that we're dealing with, they will fragment more and more into rare diseases. Rare diseases are basically all named after an enzyme or after a receptor. The diseases that we are usually dealing with, they are named after an organ, or they are named after a doctor, or they are named after a symptom. You know, the major diseases that we have heard today about and that we're dealing with are basically mechanistically not understood. I mean, heart failure, someone could have said that and diagnosed it in the 19th century. Of course, we are now more sophisticated in diagnosing it, but the way we describe it is still heart failure. Maybe HFrEF, a slight differentiation. So, to analyze the situation where we stand is most of the current drugs just target symptoms or surrogate markers. New drugs that have been introduced rather perpetuate old targets. There have been hardly any new targets or mechanisms being introduced into cardiovascular therapy. The efficacy is low, the efficacy is there. You need to treat with antihypertensives, you need to treat with statins, but you saw from the previous talk, the other data, that the efficacy is quite low, therefore the NNT is high. And the failures in drug discovery accumulate. The many failures in HDL drug discovery, of course, cost a lot of money and are a waste. We have a huge validity crisis with respect to our scientific knowledge, and our concept of disease may be a little bit outdated. If you think about it, if something goes wrong in our body, it has to be connected to a biochemical signaling mechanism whatsoever. This is quite likely to occur in several organs. It's not imaginable that there should be a set of signaling pathways that occur only in the heart, some only in the kidney, some only in the lung, some only in the GI tract. So if something goes wrong with the pathway, it's quite likely that we have symptoms in the heart and in the lung, or in the brain and in the kidney, because it's the pathway that determines where we will have symptoms. Yet our entire terminology in medicine, how we operate, how we study medicine, how clinics are organized, is there is a heart clinic, there's a lung clinic, there's a GI clinic, and so forth, as if these diseases would have nothing to do with each other. So what can big data systems medicine possibly improve? Big data is actually doable for everyone. There are so many data sources that are underused out there. So genomic data, proteomic databases, metabolomic databases, and interactomic databases, so protein-protein interaction databases. They are heavily underused. I show you one use for it, and that's a paper that I would really recommend everyone to read. I think it's one of the best papers I personally have ever read. It's from Go et al. In PNAS from 2006. This is the network of all human diseases, based on genetic links between those diseases. If a bubble is larger, it means there are a lot of genes associated with that disease. The colors of these bubbles reflect in which drawer we currently put the disease. So this light blue are all cancers, this red are all neurological diseases, metabolic diseases have a light brown, cardiovascular diseases have a darker brown, and so forth. And so these are called nodes, and the edges, the lines between the diseases, are genes that are shared between two diseases. If two diseases have the same risk gene associated, that may mean that these two diseases have something in common. And if two diseases share a lot of diseases, they merge a little bit closer together in the network. If they don't share any gene at all, they may end up in completely separate places in this network. That's how this network is generated. And you see that there are clusters. So that means some diseases have a little, or disease phenotypes as we call them, have a little bit more in common than with other diseases. It's not a homogeneous network. It's a network of little clusters. And you see some clusters that actually have one color. This one and that one. And that means that we put these disease phenotypes into one box, and that's okay. They probably belong in one box. And these are, for instance, all cancers. And here some neurological diseases, some forms of epilepsy and some rare neurological diseases where it's conceivable there are ion channels involved that really occur only in neurons. Therefore, it's a fairly homogeneous cluster. But all other clusters are pretty multicolored, which means diseases come together and really belong together that we currently treat in totally different clinics by different specialists, as if they would have nothing to do with each other. But they have a lot to do with each other. They share probably a common mechanism, and the symptoms just appear in different organs. And if we want to understand diseases mechanistically, we have to dig into these clusters. There is, for instance, cerebral, cardiovascular, metabolic disease cluster here, where a lot of the diseases that we've heard, but not all cardiovascular diseases, appear here. There is this cancer cluster, there is a cluster of retinopathies, and there's a cluster of primary immune diseases down here. And in those fields, there is the same discussion going on in the moment. So in the moment in inflammatory diseases, we have colitis ulcerosa, we have rheumatoid arthritis, we have psoriasis. And in all of these disease phenotypes, some patients, for example, respond to TNF-alpha agonists. And clinicians working in this field are now convinced that we should give up this terminology, colitis ulcerosa, psoriasis, and so forth, and look at all those patients that all respond to TNF-alpha agonists because they probably represent a mechanistically defined disease with symptoms in the colon, symptoms in the skin. But let's not define the disease based on the symptoms. The same is going on in retinopathies, where it's also showing that what used to be a homogeneous diagnosis is actually at least 3 completely different diseases with completely different pathomechanisms, some actually with a vascular component. And in cancer, we actually doing that already. What I'm proposing here for cardiovasculars and others for immunological diseases and retinopathies. Namely, when I studied medicine, we were still learning lung cancer, GI cancer, brain tumors, and so forth. No one in the clinic in the moment treats cancer according to the original organ in which the cancer occurred. It's now highly individualized. It's basically you look for the cell, you look for the expression pattern, the targets in that cell, and you treat that patient individually with the drugs to which this tumor, this cell responds. An organ-based disease nomenclature has been completely given up for cancer. And if we look now a little bit closer into the cluster of cerebral, cardiovascular, metabolic diseases, we find a lot of these disease phenotypes that we are treating in the moment more with surrogate approaches. And from those drugs here that we've listed, we find a couple of drugs that do not appear there, the targets do not appear there. But a couple of the drugs that we are using actually do appear, either via their genes or by the drugs, which may mean a differentiation between symptom-based and slightly mechanism-based drugs. So the green ones may be drugs that actually target a mechanism if we identify the patient that really suffers from that defect. And to show you also the reverse, this is actually basically this network. We're giving up the approach of targeting a single drug. You will hear more and more in future years about combination therapy, but not combination therapy of 2 or 3 drugs like in the polypill, but actually network pharmacology. If you imagine, I think it's too complicated to go into this network. It shows cyclic GMP formation, oxidative stress targets, and so forth. And the blue are all drugs that are in the clinic. But if you imagine this whole signaling network is pathologically moved, let's say, to the left, and you start getting disease symptoms, it's quite unlikely that with a single drug you will be able to correct the whole situation. So it's more feasible to say, okay, I need to block something here, I need to block something here, and I need to stimulate something here. That's called network pharmacology. And by this combined approach, you move the entire signaling network into a more healthier condition. And to show the power of systems medicine, we looked, for instance, at the target of nitric oxide, so of nitroglycerin, of these NO donors, since over 100 years, an exclusive cardiovascular target. But you may have seen that in this cluster where cardiovascular diseases are, there are a lot of neurological diseases as well. Alzheimer, for instance, and coronary artery disease share the same risk factors and may actually share the same mechanisms. So we looked at this purely cardiovascular target with systems biology and asked what would systems biology tell us about this target. So we didn't only use network-based genetic links between diseases. You can also generate the network based on symptoms. You can also generate a network based on comorbidity. If 2 diseases occur very often in the same patient, that's another evidence that diseases must have something to do with each other. And based on all these networks, you get a super network that is even more robust than a network just based on genetics. And we did this for this target and the soluble G cyclase. And then we looked, based on the protein-protein interactions, in which diseases should you look for an indication. And yes, we find cardiovascular targets, but we find a lot of neurological targets. So the same target is relevant for cardiovascular disease phenotypes and for neurological disease phenotypes. And because stroke appeared there quite often with a high rank, we did then a proof of concept study. We looked whether the target is affected in stroke. And we showed that basically in stroke, you completely lose the stimulation of this G cyclase by nitric oxide. It's completely gone. There is an alternative drug called SGC activators that can reactivate that damaged protein. If we give this protein, we reduce neurotoxicity, but more importantly, we reduce the infarct size and we reduce mortality. So it's called drug repurposing, and that's also the principle of these clusters. If these clusters are true, then basically any drug that is used within this cluster should be repurposable to any other disease phenotype within this cluster, or to most of the other disease phenotypes in this cluster, because they all share the same mechanism. And once we have a mechanism-based drug, we should be able to repurpose. And in retrograde, we can explain a lot of repurposings that have occurred in this cluster. And the same is done in all these other disease phenotypes. And the next stage, of course, is the slight limitation of this here is, of course, it's 2D. And sometimes the graph is in 2D. And sometimes you're just limited by the space on the paper. Where do I put these diseases? Because there is just no space to put it here. Otherwise, you can't see anything. So the next step is to actually show it in 3D. And also, how do we plot it? And since diseases or these mechanisms have to be linked to the interaction of all proteins, or all metabolites, all signaling networks within our body, that paper by Menche et al. Showed if you basically use the entire interactome and plot, so interactome are all protein-protein interactions, all metabolic events that go on in our cells, and plot now the risk genes that have been identified for a certain disease phenotypes. You see then basically in this entire signaling network where the site, for instance, in this case, multiple sclerosis, rheumatoid arthritis sits, and that if these linked genes actually overlap, that means that these diseases also must have something in common, and that's exactly what we also saw in the diseasome. And the next level is to plot it actually in 3D. So you find the same interaction, the same clustering, but now it's not limited by plotting it on 2D. And you see now that basically that represents the entire interactome, all protein-protein interactions in the cell, all metabolic events. And in there, you see then now where do cardiovascular diseases sit? Where do the pathways that have been identified sit? So we think this is a pretty sort of, it's non-hypothesis driven, but in the end, very precise way forward. And what we will also see, the first human genome to sequence cost over $1 billion. Now it costs 1,000 euros. And in a couple of years, it's like Moore's law, you will get your entire genome deep sequenced for probably about 100 euros in a couple of years. So everyone will get its own, his or her own personal genome. And this paper in Cell showed basically a look into the future to do this, plus measure all biomarkers that are currently available to the whole transcriptomic in the patient. And in this case, they actually did that for a year, and in that patient identified the moment where the patient had the day, where the patient had a viral infect and developed diabetes and was immediately treated for it. That's a of course, a look into the future, not feasible yet, but definitely a look into the future. So the aim is by defining diseases more precisely, not by symptoms, to leave those patients that maybe share a symptom but not the mechanism of disease, leave them untreated, and treat those patients that respond already well enough to the current drugs, which are maybe coincidentally mechanism-based with the current drugs, and those patients that you've also seen that still die because of, despite they are treated with statins, despite that their blood pressure is perfectly adjusted, treat them better by drugs that target their mechanism. So what system medicine will probably improve, but we will move from surrogates to mechanistic disease definitions, ideally with a number treated of 1. We identify the mechanism in the patient. We know if we target that mechanism, the patient will have a benefit from it. That's in an ideal world, a number needed for 1. I think we will move, that's happening in a lot of fields. Maybe cardiovascular is lagging a little bit behind in that movement. We will move to completely new disease definitions. And we will see definitely much more diagnostics and combined with drugs, therapeutics, which is called theranostics. And we will see much more sort of individualized personal monitoring. And that's a thank to the funders. Thank you.